Welcome to this explainer. Okay, so today we're talking about a term you've absolutely seen splashed across science headlines lately, CRISPR. But here's the thing, before it became this massive biotechnology buzzword, it was, and actually still is, an ancient, highly adaptive defense mechanism found in bacteria and archaea. We're going to dive right into exactly how this natural defense system operates on its home turf. I mean, think about it for a second. We are talking about incredibly tiny, single-celled organisms here. How do they possibly manage to defend themselves against these relentless viral invasions from bacteriophages? And even more mind-blowing, how do they build a genetic memory of past attacks so they can actually survive the next one?
Okay, let's dive into this. Here's our roadmap for bridging the gap between bacterial biology and human biotechnology. We'll start with the microscopic battlefield, look at the anatomy of the defense, walk through the three-step counterattack, pivot from bacteria to biotech, and finally wrap up with the challenges and the future.
So, starting off with part 1, the microscopic battlefield. There is literally an invisible war happening right now in petri dishes and natural environments everywhere. Bacteria are constantly fighting for their lives against invading viruses and genetic elements like plasmids. And to survive, bacteria use CRISPR. Now, what's really interesting about this is how perfectly it mirrors our own immune system. Where humans use antibodies to remember an infection, the CRISPR system uses spacer DNA. Where we use immune receptors for detection, bacteria use something called CRISPR-RNA. And for the actual counterattack, where we rely on immune cells, bacteria deploy Cas proteins. It's a highly sophisticated adaptive defense sequence, all happening on a microscopic scale.
Let's break down the anatomy of this defense.
So what exactly makes up this bacterial armory? Well, the first component is the CRISPR locus. You can really think of this as the bacterium's memory bank. It's a specific region of DNA that contains these short, repeated sequences, and they're separated by unique spacer sequences. And here's the wild part. Those spacers are actually derived from previous infections. They are quite literally a database of viral mugshots that serve as permanent genetic memories for the cell.
Next up, we have the Cas genes. These encode CRISPR-associated proteins, and the absolute most famous one is the Cas9 protein. If the CRISPR locus is the memory bank, Cas9 is the weapon. It acts as a pair of literal molecular scissors, and its entire job is to physically cut up foreign, invading DNA to neutralize the threat.
But you might be wondering, how do those molecular scissors actually know what to cut? Well, that's where the third component comes in, CRISPR-RNA, or crRNA for short. These are RNA molecules transcribed directly from that CRISPR locus memory bank we just talked about. The crRNA basically carries that viral mugshot and acts as a homing beacon. It physically guides the Cas scissors to the exact, precise target DNA. Let's move to part 3 and see how this all builds into the three-step counterattack. Picture the scene. A bacteriophage lands on the surface of a bacterium and tries to inject its deadly DNA, hoping to completely hijack the cell. But this particular bacterium already has a memory of this specific virus stored away in its CRISPR array. So what happens next? The immunity loop kicks in, and it works in 3 incredibly precise stages. Step 1 is adaptation, also known as spacer acquisition. If it's a brand new infection, the bacterium captures a tiny piece of the viral DNA and inserts it right into the CRISPR array as a new spacer. Boom, a new genetic memory is created. Step 2 is expression. The bacterium transcribes that CRISPR region into RNA, producing those tiny CRISPR-RNA mugshots. And finally, step 3 is interference. If that same virus attacks again, the CRISPR-RNA guides the Cas protein straight to the matching viral DNA. The Cas enzyme cuts and destroys the viral genome, completely stopping the infection in its tracks. Now, bacteria actually use a few different classes of these systems to shape their evolution, protect against phages, and even block plasmid transfers. Class 1 CRISPR is a much more complex system relying on multiple Cas proteins. But Class 2 CRISPR, it uses just a single large protein like Cas9. And it is exactly this simplicity, the elegance of this single protein Class 2 system that made it so easy to engineer, eventually turning it into an absolute superstar in biotechnology. Which brings us to part 4, moving from bacteria to biotech. This is where we dramatically pivot our explainer scope from a microscopic bacterial war to massive global human applications. Because scientists figured out how this bacterial immune system works, they realized they could program the CRRNA to target any DNA sequence, not just viral DNA. And the applications are revolutionary.
In gene editing, we can now add, remove, or modify DNA sequences. In medicine, it's driving forward gene therapy, genetic disease correction, and cancer research. Over in agriculture, we're using it to develop disease-resistant and stress-tolerant crops. And in microbial engineering, it's being used to improve biofuel production and even develop new antibiotics.
Because of its profound impact across everything from molecular biology to genetics and evolutionary biology, the discovery and application of the CRISPR system is widely considered one of the most important biological breakthroughs of the 21st century. Seriously, it's a real game changer.
Finally, let's look at Part 5, Challenges and the Future. Of course, no technology is without its hurdles. Let's take a grounded look at the trade-offs here. And this brilliantly illustrates where we are right now. On the advantages side, CRISPR is highly precise, cost-effective, easy to design, and it works efficiently in a ton of different organisms. But look at the limitations. We still face challenges with off-target mutations, meaning those molecular scissors sometimes cut the wrong piece of DNA. There are also significant delivery challenges in actually getting these systems into the right human cells, potential resistance evolving in the bacteria themselves, and, understandably, major ethical concerns regarding gene editing in humans.
But despite those challenges, the horizon is incredibly bright. Future developments are already pointing toward CRISPR-based antiviral therapies, highly tailored precision medicine, advanced synthetic biology applications, and gene drive systems. We are truly just at the very beginning of optimizing what this bacterial machinery can do for the world.
So what does this all mean? The ultimate key takeaway is this. The origin of our greatest, most advanced modern gene editing tool is simply a single-celled organism's memory bank. CRISPR is a natural, adaptive, bacterial immune system. It stores fragments of viral DNA, and it uses Cas proteins to recognize and destroy invading genetic material.
It really makes you wonder about the natural world, doesn't it? If a microscopic war in a puddle of water gave us the key to rewriting DNA, what other brilliant microscopic defense mechanisms are currently hiding out there in the microbial world, just waiting to become tomorrow's next massive biotechnology breakthrough? Thanks for joining me on this explainer, and keep questioning what's under the microscope.